I'd like to thank Chip and the Foundation once again for this opportunity to present. I've learned a lot from all of you for the last couple of days. And so here I'll talk about how to interpret ADA2 variants. I have no conflict of interest or disclosures related to this talk. So I will first make the argument that proper interpretation of ADA2 variant is a very important thing. Right? So as a pediatric rheumatologist, we're now sending more and more sequencing panels for patients with possible autoinflammatory or autoimmune disorders. Now, ADA2 is already included in most primary immunodeficiency as well as periodic fever syndromes that we send. So we often are getting now variants of ADA2 coming back that requires proper interpretation. There are to date more than 90 DATA2 associated mutations in ADA2 that's been described. And there are many more polymorphisms that have not really been studied. In addition to that, not all patients have two identifiable mutations. And if a patient comes up with one mutation, it's really important to know whether that one is pathogenic or not, and look for the other mutation if needed. Furthermore, there are carriers of one mutation that may be symptomatic. So again, it's really important to know the status of those mutations. So as a rheumatologist, my view of genetic variant is quite limited. I think of variants in one of two categories. You have benign variants, which are polymorphisms, right, found in healthy people. And then you can have pathogenic variants, which affect gene function, right? In this case, it causes DATA2. When we find a variant of unknown significance, to me, that means it's something that we just don't know what it does yet, right? We don't know which category it belongs. But once we sort it out, it would go into either one of these baskets of either a benign variant or a pathogenic variant. But in the views of a geneticist, this is much more complicated. This is a snapshot from the American College of Medical Genetics guidelines and really how to interpret a sequence variant that we see. They go through a very complex set of criteria, right, that's dependent on a number of factors, including population data, computational predictive data, functional data, and also sex segregation data, as well as allelic factors. And within each of these categories, you can look for different levels of evidence, going from very strong to supporting evidence to the mutation being a pathogenic variant or a benign variant. Then based on different boxes you check here, you go through a table of how many boxes you checked and which box you checked. Then you classify the variant as pathogenic, likely pathogenic, benign, likely benign, or we still don't know what it does in terms of uncertain significance. So when I see a variant in ADA2, this is how I think about it. And certainly there are many ways to think about this. There's no right or wrong way, certainly, but this is sort of one approach. So first I want to know about whether this particular amino acid affected by the variant or the region have been reported in previous DATA2 cases. You can find this information fairly readily in two places. One is in the NCBI ClinVar website, where they keep track of patients with mutations affecting various genes. And then the InFevers database is also very valuable here. It's frequently updated with the most up-to-date list of ADA2 variants, whether they're pathogenic or not. Next, I want to know how common is the variant in the general population. And you can find this information in the NOMAD database from the Broad Institute, where they have genotype data from over, I think the orders of 140,000 individuals. Next, I want to know if the variants predicted to be pathogenic by in silico algorithms. And many of you probably have experience of this, where there are a lot of different algorithms out there, right, that can predict the impact of a sequence variant, typically missense variants, on the gene in terms of function. So they're based on the impact on structure, evolutionary conservation, the substitution of amino acid properties, and also some combination of these methods. So there are many out there that you're probably well aware of, like the CAT score, SIFT, PolyFin, and many more. And lastly, are there functional data available to look at the specific variant of interest? So this is what we can add to this topic. In the lab, we've been doing functional evaluation of DATA2 associated mutations. So this is done by expressing different variants using a expression construct in 293 T cells, and then measuring the output of ADA2 activity in the supernatant using a spectral photometric assay from Dr. Michael Hirschfield's lab. So far, we have analyzed more than 70 of these mutations associated with DATA2 in the literature, 51 missense mutations, a few single amino acid deletions, and a number of insertion deletion with frame shift, and several nonsense mutations as well. So on the bottom here, we can see on the y-axis is the retained ADA2 activity as a percentage of wild type. We can see here the missense mutations seem to have a fairly wide range. There are many hypomorphic missense mutations that retain activity as far as high as the 60-65% range, and also many that have some residual activity in the 10-20% range. Versus the insertion deletions and nonsense mutations have no residual activity at all, as you may expect. Plotting different missense mutations on a diagram that goes through the different domain, going by amino acid order, we can see these hypomorphic mutations in ADA2 seem to scatter through various domains of the gene without specific co-localization or clustering. We've also done some in silico analysis of the different algorithms for these DATA2 associated mutations. And again, these are mutations that have been found in DATA2 patients based on publication from many of you. So on the y-axis here, these are different mutations that we've collected from the literature. And on the x-axis is the different in silico algorithms we use to analyze. And each of these algorithms will help predict whether the variant is pathogenic or not, right? So if it's the green box, that would be predicted as benign. And if it's orange, then it's predicted as pathogenic. So you can see overwhelmingly that these algorithms work quite well. Right? Most of these boxes are orange because we expect them to be pathogenic based on finding in patients with a particular phenotype. For example, CAT score, which is widely used as a prediction of pathogenicity, it almost predicted every single mutation with the exception just a few correctly. There are some interesting patterns, though, that a lot of these algorithms did not predict correctly mutations in the N-terminus, which I think is attributed to variation in the N-terminal signal peptide between different species that's not really well conserved. So can we use a similar approach of functional analysis to study different variants of unknown significance in ADA2? So to do that, we first gather a list of ADA2 variants from the NOMAD database constructed by the Broad Institute. So this is fairly easily accessible through the website from NOMAD. And we look for the ADA2 gene and then sort it out by the allelic frequency, looking specifically at missense mutations. So we looked at the top 100 most common missense variants on the list. We made expression constructs for each one of those, just like we did for the pathogenic mutations. And the allelic frequency of these mutations range from very high, from 0.34, basically 1 in 3 people, right, to 0.000012, which is about 1 in 80,000 people. By analyzing this list, we incorporated about 99% of the allelic variants by allelic count in the NOMAD database for the ADA2 gene, at least for the missense variants. And if we exclude the most common polymorphism, histidine 355 arginine, which is seen in 1 in 3 people, a very common polymorphism throughout the world, then we're still covering about 93% of the other variants. So we feel like this is a fairly good coverage by analyzing the first 100 variants on this list. So what do we predict by doing this type of analysis? Or again, we think about sorting the variants into a category of benign versus pathogenic mutations. And one possibility is that when we look at their ADA2 activity, we can see a cluster of benign mutations, right, that have very high retained ADA2 activity. And then we have a cluster of pathogenic mutations that have very low or absent ADA2 activity in 2 distinct categories. The other possibility is we may get a full spectrum of findings where different variants affect ADA2 activity or not, right, in variable ways. So on the left here, you have a number of mutations that are fairly benign, right, with very little impact on ADA2 activity, to very pathogenic mutations, right, that have no retained activity at all. And most of these variants would be falling somewhere in the middle of the spectrum. So in fact, when we did this analysis, we saw the latter possibility. So this is a diagram with 100 bars. It's too busy to put individual variants on the bottom here. But we can see that it really forms a nice spectrum. So this is the retained ADA2 activity, right, from the expression of the 100 variants in ADA2, going from many that have no impact at all, right, near 100% of wild type ADA2 activity, all the way down to the bottom here where we have little activity remaining. So this is again ranked by the residual activity. So on this end, this would be the pathogenic mutations. Sorry, this end would be the benign mutations. On the right end would be the pathogenic variants. Right. So this brings up an interesting question, right? How much reduction in ADA2 activity do we call it, DATA2 associated? Do we call it truly disease associated in terms of its pathogenicity? So this question could be in part answered by looking for variants in this list that have previously been seen in DATA2 patients. Right? So these are simply the top 100 most common ones, but some of these have been described in patients before. So of the list of 100, there are 21 variants that have been found in DATA2 patients previously. So all of these are highlighted in the red bar. The first thing you notice is that there's a heavily skewing on the right side here, which makes sense, right? Which, a lot of the variants with low ADA2 activity, they have been described in DATA2 cases because they're really pathogenic. What's also interesting is there are a number in the middle here, right? These are the hypomorphic mutations, right? Even as much as 60% residual activity have been seen in patients with DATA2. So we don't really need mutations to be completely disruptive of gene function, right, to cause the disease. When we compare the DATA2 associated variants with the other variants, which we at this point call variants unknown significance, of course, DATA2 variants have much lower ADA2 activity compared to this control group. And another interesting finding here is by plotting the allelic frequency of these variants with the ADA2 activity compared to wild type. There's a general notion that if the variant is rare, right, that is more likely to be pathogenic. And we see that it's actually not really that true. If you look on the right, so there are two variants. These are the most common variants for the gene. So H335R, again, is found about 1 in 3 people. And V349 isoleucine, this is one found about 1 in 400 people. So these two most common variants have essentially normal ADA2 activity, right, near 100% of wild type ADA2. But beyond that, we don't really see any correlation between among the 100 variants that we analyzed between the allelic frequency or the amount of ADA2 activity that's remaining. So next, we perform in silico analysis using the different algorithms. Right? So are these algorithms good in terms of predicting whether ADA2 variants are that have high ADA2 residual activity or low ADA2 residual activity? There are certainly many parameters we can use to do this type of analysis. So we choose to do a receiver operating characteristic curve for each of these and calculate the area under the curve. So the ROC curve allows a summarizing characteristic of both sensitivity and specificity for each of these methods. And looking at all of these different algorithms, we find the ones with the highest area under the curve are those seen here in the red color. So CAT score is really good, DAN score is quite good, and also a number of other commonly used algorithms, so CIF score, Polyfan. So all of those are all up there in terms of its area under the curve and ROC analysis. On the graph down here, this is plotting out some of the ROC curves. And you can see here that most of these have essentially overlapping curves in this type of analysis, showing you both good sensitivity and specificity in predicting whether variants are benign versus pathogenic. On plot C, I would show you an example where if you plot the actual raw score generated by the CAT algorithm, right? So a high CAT score would be predicted to be pathogenic. A low CAT score is predicted to be benign. You can see those with mutations on the x-axis with very little ADA2 activity remaining. Right? They tend to have very high CAT score, right? Basically matching up with the prediction that those are likely to be bad. And mutations that have close to normal ADA2 activity tend to have lower CAT score in general. So the two N work quite well in forming a line here. What gets a little tricky is the mutations in the middle where they're hypomorphic. So some of them are predicted to be benign, some of them predicted to be pathogenic based on the CAT score. So these algorithms work well on both extremes, but the ones in the middle, as you would expect, may be difficult to tease apart. And the same concept goes through other scores as well, in terms of DAN score, the best 3, and CIF analysis. So just show you a few here without going through too much details. But that's all nice, right? It's all in vitro, it's all functional work using the cell line. Right? Can we use this information, right, to predict ADA2 levels in human? And we can think about doing this in a couple of ways. So first, we can think about doing this in DATA2 patients, right? So looking at plasma ADA2 activity in DATA2 patients with biallelic mutations. This is a little bit difficult because activity in these patients typically is very low or even absent. And with that, it's really difficult to assign impact to specific alleles. The other way to do this, which we've done, is looking at plasma ADA2 activity in carriers with one ADA2 mutation, which you can, to some extent, infer what the impact of that one mutation is on the plasma activity. So shown here is a graph that we have done looking at ADA2 activity in the plasma for 41 individuals with a variant of ADA2. First, let's look at the background lines here. So the line up here is the median level for healthy controls. The second line here is a lower limit for healthy controls. And the bottom one here is typically the limit under which we see patients with data 2. So I want to acknowledge Dr. Michael Hirschfield for teaching me how to do this plasma assay. And this is also done in collaboration with Dr. Ivona Exentivich at NIH. So looking at these dots, each dot, it's in the color codes for a specific variant of ADA2. Some of these you can recognize as no mutations in the gene. So let's start from the left. So on the left are these white circles. These are patients, carriers with a null mutation. So these are early termination, nonsense mutation, or frame shift indels that predicted to be loss of function mutations. So these patients uniformly had carrier level range of ADA2 activity under the lower limit of healthy controls. And that's similar to for patients with mutations that are predicted to have very little residual function in our functional analysis. So R169Q, for example, we predict to have less than 1% of the activity of wild type. So all the carriers of R169Q tend to be in this carrier range as well as we predict. As we move over a little bit to the right, where we see some mutations that are more in the hypomorphic spectrum. So G47R have about 10% residual activity, and G47A have somewhere in the range of 15 to 18% residual activity. We can see for these variants, the carriers of these particular variants have a sort of more widespread level of ADA2 plasma activity. Some are in the carrier range, but some are in the low normal healthy range. Moving further to the right, we see variants that are predicted to be hypomorphic, but with more retained activity in our functional analysis. So F355L have somewhere in the range of 50 to 60% retained activity. And those you can see that carriers, these variants tend to have higher activity now closer to the healthy control range. There are a few variants, individual few variants that we found, where these are variants that predict to have no impact on ADA2 function. So these, for example, H335R found in 1 in 3 people, and D373V as well as E49Q that have really minimal impact on ADA2 function in vitro at least. And all of these carriers tend to have ADA2 plasma activity in the normal healthy control range, slightly above the median of healthy controls. And so looking at this, there's a general trend. You can see that the ADA2 plasma activity in these carriers seems to match in part, right, to what we predict in vitro. But of course, there's plenty of variability in this type of study, right? Even the same mutation for G47R, for example, there's a fairly wide range. Now, what can we do with this functional data from all of these variants? Right? So of course, we can provide complementary data to the plasma activity assay, as we showed in the last slide. We can give you a reference to distinguish potentially pathogenic mutations from benign variants. And knowing sort of a full list of pathogenic or potentially pathogenic mutations, we can use the information to help better calculate the carrier frequency and disease prevalence. This is done in collaboration with Dr. Ann O'Donnell and Sam Baxter from the Broad Institute, who have already presented on this work. And also, this may allow us to look at the impact of ADA2 variants associated with other diseases. So what does that mean? We're talking about data too, but what other diseases can we talk about? So there's some interesting concept in Behçet's disease that we can discuss here. So in some data 2 patients, right, they have features of Behçet's. Certainly, sometimes you can see oral ulcers and some evidence of vasculitis that we've seen in Behçet syndrome. And this interesting study where they looked at 355 individuals with Behçet's disease. And looking at their different genetic variants, they found a number of individuals with variants in ADA2. So they're seen down here. Right? So they found 6 different variants of ADA2. So some of these, of course, are very common. Right? H335R, again, 1 in 3 people. V349I, it's found in 1 in 400 people. Right? So the expected frequency of these variants came out quite, on target. Right? So we expect 234 people for this variant based on the allelic frequency. They found 218 people, where about 3 people for V349I. But then they also found several individuals with rare variants that we don't expect to find in such a small cohort. Right? So these are A247V, R49W, and T360A. Right? We don't expect to find these in 355 people based on very small allelic frequencies. But they found 5 people with these variants, and also some people with a 309 isoleucine variant. And what's interesting is all of these variants have, at least in part, a reduction of ADA2 activity compared to the wild type ADA2 in our functional analysis. So these are likely to be hypomorphic variants that are found in patients with Behçet's disease. T360A is a known mutation published by Dr. Cortano's group. Also interesting, if you look at these variants using the different algorithms I talked about earlier, most of these should be predicted to be benign, right, and not pathogenic, with the exception of T360A, which have been found in DATA2 patients. So again, this information on the different variants allow you to look at, to perform analysis and look at the impact on ADA2 variants, both in DATA2 and also other conditions. So with that in mind, I'd like to conclude that so that we can estimate the impact of ADA2 variants on protein function, right, by analyzing this using this assay of ectopic expression and enzymatic activity measurement. We found that ADA2 variants can show a wide spectrum of impact on its residual enzymatic function based on analyzing the top 100 most common variants in the GNOMAD database. We found that the in vitro data roughly correlates with plasma activities in a number of carriers. And this data set potentially provides a reference to distinguish potentially pathogenic versus mutations in ADA2 versus benign variants. So we're working on ways to get this information to the community. In the meantime, if you have any questions about any variants you have found, and when we have done the analysis, just shoot me an email. We're happy to share any of the data. So I'd like to acknowledge people in the lab and also a number of collaborators seen here. So thank you very much for your time.